CRISPR-based gene editing represents one of the most transformative technologies in biomedical science, offering an unprecedented level of precision in manipulating the human genome. Among its many experimental applications, one of the most intriguing is its potential to regulate genes involved in telomere biology, specifically those controlling the enzyme telomerase and associated cellular machinery. Telomerase, composed of the catalytic subunit hTERT and the RNA template HTR, is responsible for extending telomeres, and its reactivation in somatic cells has long been considered a key to delaying cellular senescence and extending the health span of tissues. By using CRISPR to upregulate or modulate these components, scientists are exploring new ways to reverse telomere shortening and restore cellular vitality at a foundational level. The most direct approach involves CRISPR-mediated upregulation of the hTERT gene, which encodes the reverse transcriptase component of telomerase. In normal adult somatic cells, hTERT expression is silenced after early development, leading to progressive telomere shortening with each cell division. Reinstating hTERT expression using CRISPR activators like dCas9, VP64, or other CRISPR tools has already shown promise in experimental models. Studies in human fibroblasts have demonstrated that activating endogenous hTERT can not only lengthen telomeres, but also restore replicative potential and reverse markers of cellular aging. This level of precision, turning on a gene without integrating foreign DNA, offers a safer and more physiologically compatible alternative to earlier viral vector-based gene therapies. Beyond hTERT, CRISPR is also being used to study and potentially edit other regulatory genes that influence telomerase activity and telomere stability. This includes genes encoding shelterin complex proteins like TRF1, TRF2, and POT1, which help maintain the structural integrity of telomeres and prevent them from being misidentified as DNA damage. Fine-tuning the expression or function of these genes may enhance telomere protection without the need to fully reactivate telomerase. Likewise, modifying genes involved in oxidative stress responses and DNA repair, such as those in the p53 or ATM pathways, may create a more favorable environment for telomere maintenance. The combinatorial power of CRISPR allows researchers to test and optimize multi-gene interventions that could mimic the natural telomere-preserving environment of stem cells. One particularly exciting development is the use of base editing and prime editing techniques, which allow nucleotide-level corrections without inducing double-strand DNA breaks. These advanced CRISPR systems are being explored to precisely tweak regulatory sequences in the hTERT promoter or other telomere-related loci. This could potentially enable long-term control over telomerase expression, either through promoter demethylation or insertion of transcription-enhancing motifs. Such fine-grained control opens up the possibility of safe, tissue-specific telomerase reactivation, especially in regenerative medicine applications, such as restoring the function of aging heart, brain, or immune cells. Despite the promise, several significant challenges remain. Chief among them are the potential oncogenic risks associated with telomerase reactivation. Telomerase is active in over 85% of human cancers, and its uncontrolled upregulation can allow malignant cells to evade the normal limits of replication. Thus, any therapeutic use of CRISPR for telomere extension must be carefully balanced with tumor suppression mechanisms. Strategies like inducible or transient activation of telomerase, or restricting editing to short-lived or regenerating cell types, may help mitigate this risk. Additionally, robust off-target screening and editing specificity remain essential, though improvements in CRISPR fidelity and delivery systems continue to reduce unintended genomic changes. Ethically, germline editing of telomere-related genes raises questions about heritable changes in aging biology. While current efforts are focused on somatic interventions, the implications of permanently altering aging pathways at the genetic level demand thoughtful oversight. However, somatic CRISPR therapies for age-related disease or tissue regeneration are generally viewed as more acceptable and are moving steadily toward clinical testing in other areas of medicine, providing a roadmap for telomere-based applications. In conclusion, CRISPR-based editing of telomere-related genes is still in its experimental infancy, but holds extraordinary potential. It offers a level of biological precision that could allow safe, controlled enhancement of telomere maintenance, pushing the boundaries of what's possible in anti-aging and regenerative medicine. As technical hurdles continue to be addressed and ethical frameworks evolve, CRISPR may one day become a cornerstone tool for not just treating age-related disease, but actively reversing molecular aging itself. The future of telomere science may very well be written one edited base at a time.